Okay, let's dive into this. Welcome to today's Explainer, where we're shining a massive spotlight on the ultimate biological buddy cop duo, DNA and RNA. If you've ever found yourself wondering how the sheer, mind-boggling complexity of life is managed, archived, and, well, actually built, you are in exactly the right place. Today, we're not just reading biology facts. We're stepping inside a microscopic cellular factory to meet the fundamental architects that have been working side by side since the literal dawn of life. So, here's our game plan for the tour. First, we'll meet the architects themselves. Then, we'll contrast the blueprints with the contractor. From there, we'll track the RNA travel fleet, break down the overarching rule of the central dogma, and finally, look at some mind-blowing real-world medical breakthroughs. Let's get moving. Section 1. Meet the biological architects. It's time to introduce the two central characters of our cellular factory, who literally keep this entire system running. First up, we have deoxyribonucleic acid, though we all know it simply as DNA. I want you to think of DNA as the ultimate sturdy archivist. As a master blueprint, it's a highly secure biological library. Its main job, its prime directive, is to safely store every single piece of genetic information required for a cell to function. It makes sure the instructions for life can be reliably passed down from one generation to the very next, just sitting safely in the nucleus, holding on to the permanent records. But, a blueprint doesn't just spontaneously build a building on its own, does it? Nope. And that is exactly where ribonucleic acid, or RNA, steps in as the dynamic, versatile contractor of the cell. If DNA is the executive sitting in the corner office with the permanent archives, RNA is the agile field agent out on the construction site. It grabs those stored instructions and actually turns them into the physical machinery of the cell, our proteins. Moving to Section 2, Blueprints versus the Contractor. Now, what's really interesting about this slide is how their wildly different roles in our factory dictate their entirely different physical and chemical structures. See, DNA is built for the long haul. It's built for stability, twisting into that iconic, robust double-strand helix. It's like a winding staircase, with two chains wrapping around each other just to protect the nested bases inside. And its backbone is made of a sugar called beta-D2-deoxyribose. RNA, on the flip side, is the ultimate pragmatist. Operating generally as a single strand, it has the incredible chemical flexibility to fold back onto itself into these complex, funky shapes like hairpins and loops. Plus, its sugar is beta-D-ribose. Because that sugar has one extra hydroxyl group attached to it, RNA is chemically far more reactive and ready to work than its sturdy partner. And then, we have the classic biochemical distinction hiding in their nitrogenous bases. Both DNA and RNA share adenine, guanine, and cytosine. But where DNA uses thymine to pair it with adenine, RNA completely swaps it out for uracil. This subtle little chemical signature is a total game changer. It dictates exactly how they interact with all the other machinery in the cell. Section 3, the RNA travel fleet. So because DNA is so incredibly massive, precious, and structurally rigid, it almost never leaves the safety of the cell nucleus. It literally can't do the heavy lifting of protein assembly itself. Instead, it relies entirely on the RNA travel fleet to get out there and execute the plans. Seriously, just look at how brilliantly the contractor delegates the work. RNA takes on 3 highly specialized forms to get the job done. First up, we have messenger RNA, or mRNA. This is the courier that physically carries the genetic message right out of the nucleus. Next is ribosomal RNA, or rRNA, which actually makes up the bulk of the ribosome itself. You can think of it as the physical factory floor where all the assembly happens. And finally, transfer RNA, or tRNA. This is your targeted delivery specialist. It fetches specific amino acids and hauls them to the ribosome to perfectly match the code that the mRNA brought over. It is an absolute masterclass in cellular logistics. Section 4, the central dogma. So the crucial point is that this partnership isn't just a random bunch of chemicals bumping into each other. It's a highly coordinated, flawlessly perfect system that governs all life as we know it. Scientists call this the central dogma. Let's define this with the scientific gravity it totally deserves. The central dogma of molecular biology is the fundamental principle that DNA makes RNA and RNA makes the proteins. It is the absolute core of cellular operation. As our source material beautifully puts it, without DNA, there is no plan. But without RNA, the plan never leaves the paper. You absolutely fundamentally must have both. Let's move to and see how this builds in a precise chronological sequence.
Step 1 is replication. This is where DNA meticulously copies itself, so every single new cell gets a complete set of the master instructions. Step 2 is transcription. DNA serves as the direct template to create RNA. And finally, step 3 is protein assembly. This is where our RNA fleet translates that newly minted code to actually build the proteins, the very machinery of life.
Which brings us to a massively important question. We've spent this explainer zoomed way into the microscopic world, looking at ancient biochemistry. But how is this microscopic, ancient machinery saving human lives in the macroscopic world today? That brings us to Section 5, Real-World Medical Breakthroughs. To answer that question, we have to look at how modern science has tapped directly into this biological factory.
And this brilliantly illustrates exactly why understanding this explainer matters so much. Scientists have moved way beyond just observing RNA's natural role. They've actively harnessed it. mRNA technology is currently at the absolute forefront of modern medicine. By utilizing mRNA's natural, everyday ability to act as a messenger, researchers are creating vaccines that deliver life-saving instructions directly to the human immune system. We're literally teaching our bodies how to fight formidable viruses like SARS-CoV-2 and increasingly even targeting certain types of cancer. We are essentially using the cell's own contractor to build our bodily defenses.
It brings our entire buddy cop narrative to a wonderfully respectful close. The steady, reliable DNA admits that RNA has kind of become the showstopper in the human world lately, while RNA acknowledges that without the safe, permanent records kept by DNA, none of it works. Together, they truly do keep the engine of life running. Which leaves us right here. We now know exactly how the biological library and the cellular contractor work together. We've seen how scientists have hijacked this very system to protect human health. So, if we can now successfully program RNA to fight SARS-CoV-2 and cancer, what other biological blueprints will we rewrite next to cure the seemingly incurable? Just think about the sheer possibilities locked inside that cellular factory. Thanks for learning with me today. I really hope this sparked your curiosity to keep exploring the incredible future of mRNA technology.